Bob, go ahead. Go ahead. Bob's going to say something really quickly.
Good afternoon. Welcome to the Blueberry session. Before we get started, I wanted to make a pitch. Black Tuesday shopping for you guys that didn't get it all done yesterday. This is the new version of the Compendium of Blueberry, Cranberry, Lingonberry Diseases and Pests. It just came out. It's $150. Well, that's just, let me finish. From now until December 15th, I can get it for half price. So if you place an order now, I will order them, and they're going to be $75. So, and I haven't read the whole thing, but the virus sections are really good. I haven't read the rest of it. If you're interested, we'll get a sign-up sheet, and people can sign up for it, and I will order them when I get back to Corvallis. Yeah, if you want to get your someone special a Christmas gift, this is the one to get. Better than flowers.
He's going to introduce you.
All right. Hello, hello. We'd like to thank Preferred Freezing for sponsoring our lunch today. It's delicious. Give them a round of applause. I'd also like to introduce Dr. Lisa Wasco DeVetter, who is an assistant professor in berry crops at WSU's Northwest Washington Research and Extension Center in Mount Vernon. Her current work focuses on physiology and sustainable production of berry crops in the Pacific Northwest. Thanks.
Well, as Chris is getting this set up for me, thank you, Colton, for that nice introduction. Again, I'm Lisa Wasco DeVetter, Assistant Professor of Small Fruit Horticulture, and I've been an Assistant Professor of Small Fruit Horticulture with Washington State University for 2 years. And this presentation, an update on some soil-borne disease management and fumigation trials, represents just a small snippet of some of the research that has undergone since the start of my program. Before I delve in any further on the information in this particular presentation, I want to acknowledge several individuals that make up what I consider the soil-borne disease management team. Several of them are here and are giving presentations, so I highly recommend you attend their presentations if this topic is pertinent to you. First, we have Dr. Inga Zazada. She is a plant pathologist specializing in nematology. And then we have Dr. Jerry Weiland, another plant pathologist specializing in fungal pathogens as well as oomycetes. Both are located in the Oregon USDA Horticultural Crops Research Unit in Corvallis. On the bottom, we have Dr. Tom Walters, who is a private researcher or private ag specialist who has a great history of doing field trials with soil-borne disease management. He's a member of the team. And last on this slide, we have Mr. Sean Watkinson. He is a technician in my program. So the goal of today's presentation is to provide just an update on some of the current soil-borne disease research in red raspberry. And I'd like to give a little bit of an outline before we begin. So I'll go and provide a brief background just to recap why we're interested in soil-borne diseases. And I'll talk just about two projects. One is a root and crown inoculum removal project, and another one is an alternative fumigation strategies project. And I'll just touch on some of the future projects or activities that we're either doing or hoping to do, pending on funding, and leave time for some questions and discussion. So let's just go into the background. So improved soil-borne disease management is a research priority. It's often articulated as a top-tier research priority by our various grower commissions, including the Washington Red Raspberry Commission. Growers primarily pre-plant fumigate to manage several pathogens. First is root lesion nematode. That's oftentimes cited as the primary reason growers are fumigating. And root lesion nematode is Pritolenchus penetrans, or RLN for root lesion nematode. You'll see that abbreviated quite a bit in this presentation. It's a migratory endoparasitic nematode that feeds on raspberry roots as well as others. It has a very broad host range. And the top image is a picture of root lesion nematode. The next one is Phytophthora root rot or Phytophthora rubi. This is an oomycete that's more problematic on heavier clay soils with poor drainage. And there's others that are of concern by growers, verticillium being among them. And Dr. Weiland will be talking about verticillium in Northwest red raspberry later on today. So despite fumigation, soil-borne disease management is still a challenge for raspberry growers. And there's many reasons that we suspect that is the case. First being that there's been changes in land management practices. So as the raspberry industry has grown up here in Northwest Washington, prime land suitable for red raspberry production has gone limited, and we're seeing less crop rotation that might help break up disease cycles and more replanting. We've also experienced a loss of some of the traditional soil fumigants like methyl bromide. With the re-registration eligibility decisions, we have increasing restrictions on fumigation usage, such as buffer zones being among some of the challenges that growers confront. There's also questions that from a research side we often have that maybe the fumigants that we are applying, we need to optimize our method of applying them, and that there's progress to be made on how we apply them. And then there's also questions about replant disease complex. And replant disease complex, if you're not aware, this is reduced growth and vigor of a planting after it has been replanted in the same site of a same or closely related species. And this can be due to biological or possibly non-biological or abiotic factors. But trying to understand this a little bit more could be very important and help growers and agriculture professionals decide how do we target and what we utilize to target particular pathogens in a specific site. So one of the primary goals of my program, the Small Fruit Horticulture Program, it's a shared goal among many other researchers that work with my program, is to develop improved horticultural tools that are practical for soil-borne disease management in the red raspberry industry. And this is, as mentioned, a shared goal among many other researchers and offers an excellent opportunity to collaborate where we can all provide our own individual expertises to try to tackle these pretty complex problems. So in my program, we've taken a diverse approach in looking at this in red raspberry. We're looking at application and management of cover crops, both in a pre-plant and a post-plant situation. We're also trying to get a better understanding of the soil microbial ecology that can contribute to that may contribute to replant disease. And then the two projects that I'll mention are the root and crown inoculum removal project, and then looking at alternative fumigation, fumigants and methods of fumigation. So I'll start off and talk about the root and crown inoculum removal. This is a project funded by the Northwest Center for Small Fruits Research and the USDA Critical Agricultural Research Extension Center program. So if you're not familiar with this project, you might be asking, why root and crown inoculum removal? Well, this came from the observation that in a pre-fumigation situation, there is a large amount of root and crown material remaining in a field. And this could be a potential source of inoculum of soil-borne diseases, because a lot of the fumigants that we're utilizing are unable to penetrate into that tissue to kill it. So this material could serve as a source of inoculum. And our question was, if we are able to remove this root and crown material in a field prior to fumigation, could we improve soil-borne disease management? So this is a picture of a field where we had one of our trials at, and just kind of gives you an idea of the amount of plant material that remains in a field. So this would be tilled in, and the field would be prepared like a seed bed before fumigation, and once the soil has achieved an appropriate moisture level. So the goals of this project, just short term, is to evaluate and look at the efficacy of root and crown inoculum removal as a pre-plant management technique for enhancing soil-borne disease suppression. We're also looking at the efficacy of this tactic as well. And then long term, it just plays into the overall goal that my program has, as well as other researchers, in terms of developing integrated and improved practical management tools for soil-borne pathogens and pests. And there are 3 experiments that I'm going to quickly go over in this short 25-minute presentation. So the first experiment that I want to highlight is looking at the speed and efficacy of different devices that could be utilized for root and crown inoculum removal. And so we tested these 3 devices here in 2 commercial fields. We looked at a Lundeby plant lifter, which is pictured here on this slide, a beach cleaner, and a potato harvester. And the reason we looked at the first 2 devices is because they're devices that are present here in Whatcom County. The beach cleaner is something that is traditionally not present up here in Whatcom County, but we had a grower cooperator purchase this material to test this hypothesis of root and crown material removal and enhancing disease suppression on his own farm. So a quick recap of the results that we have from this experiment. When we looked at the speed and efficacy of root and crown removal, we found the potato harvester was the fastest, covering about 1 mile per hour, whereas the plant lifter was the slowest at a painstakingly slow 1/4 mile per hour. But that plant lifter was also the most effective, removing about 98% of the plant material within a 4 cubic foot volume of soil, whereas the beach cleaner was the least effective and the potato harvester was moderately effective. And this just gives you a visualization. So when we excavated a 4 cubic foot area, this was the amount of material that was lifted and removed by the plant lifter that would have been removed prior to pre-fumigation. And this is what was remaining when we sifted through that volume of soil. So again, it was pretty effective at removing this material. So now on to the second experiment. The objective of this experiment was to look at the effects root and crown removal has on root lesion nematode, Pythium, and Fusarium, as well as raspberry plant growth, development, and yields. This was initiated in a field of Shemanis that was being replanted to Meeker, and it started in August of 2014 up here in Whatcom County. The experimental design, or the approach that we used, we had a split plot experiment where our main plot was fumigation with or without using Talone C35, and the split plot was root removal with or without with our Lundeby plant lifter. And this design allowed us to test all these combinations of fumigation, no fumigation, root removal, and no root removal. It was replicated 6 times, and the main plots were 100 by 30 feet, and the split plots were 15 by 30 feet. So pretty large size plots. And the data that we collected include changes in populations of soil-borne diseases, root lesion nematode, and Fusarium and Pythium. We did not collect data on Phytophthora because that site didn't have a high pressure of Phytophthora, so we looked at Fusarium and Pythium as proxies for this pathogen. And we also monitored plant growth and yield. So I presented on this project last year, so 2015 results, here's a short recap. What we found was that colonization rates of the root material that was greater than 5 millimeters in diameter by Fusarium and Pythium was the same 6 weeks after fumigation. So 6 weeks after we fumigated in August, colonization by these organisms was not impacted by fumigation. Soil populations of Fusarium and Pythium were the same in the bulk soil by spring of 2015, so prior to planting our plants in the field. Root lesion nematode populations, they returned to pre-fumigation levels by August of 2015, so about 1 year after. They were the same across all treatments at that time point, and we didn't see any differences in plant growth in 2015. And here's what we learned in 2016. This is some of the plant data that we collected. So we looked at primocane number, primocane height, primocane diameter, yield, which was the average of 4 machine harvests, and then average berry size. What we see at the statistical level is no significant difference across the treatments, but numerically, there's a pretty consistent trend that our fumigated plots have more growth and higher yield relative to our unfumigated plots. And we're really not seeing any benefit in this trial due to root removal. Over time, when we look at the data all the way through September of 2016, we did see that fumigated plots had less root lesion nematode than our non-fumigated plots when we looked at the populations in the soil or in the roots. There were no differences in Fusarium and Pythium populations, and we did see some pretty striking differences in the vigor of the plantings within the different plots. So this image here is supposed to give you a representation of that. In our fumigated plots, we had much more dense canopies and lush growth relative than our non-fumigated plots. We didn't see any benefit or difference with root removal. And then experiment number 3 was very similar to experiment number 2, but the field site that we were working in had a much higher and consistent population of Phytophthora rubi. So we looked at the effects of root removal on Phytophthora rubi, Fusarium, Pythium, as well as root lesion nematode. Although this particular site does not have a really high population of root lesion nematode. And we're also tracking plant growth, development, and yields. This project was initiated in the field of Meeker that was being replanted to Meeker in August of 2015. And the root removal device that we utilized was the beach cleaner. So the design, real quick, was a randomized complete block design where we had 2 treatments with and without no root removal. This was replicated 4 times in plots that were 30 feet across, so covered 3 rows and were 60 feet long. And the entire field was broadcast fumigated with Talone C35. So we did not have the non-fumigated check for this experiment. And the data that we collected were similar to experiment number 2, Phytophthora, Fusarium, Pythium, root lesion nematode, plant growth, and we'll be looking at yield in 2017. So a recap of some of the data or what we've learned so far in this particular experiment. We haven't seen any differences in prime decay number, height, diameter, nor vigor within the planting. Fumigation did not reduce Fusarium and Pythium populations in large and small root debris. Within the bulk soil, we found that fumigation initially reduced Fusarium populations, but not Pythium. And this was about 6 weeks after fumigation. And then we're still generating the Phytophthora data. This takes a little bit longer to generate. Root lesion nematode, we really saw a few populations of root lesion nematode for this particular site. But again, the theme is we didn't see any effect of root removal for this experiment, at least at this stage. So the take-home messages for project number 1, what we've observed is that root and crown inoculum removal is not effective in the current fumigation systems. And I'd like to underscore in the current fumigation system. So what we're learning is that broadcast fumigation with Talone C35 untarped is really leading to variable to poor suppression of root lesion nematode, Fusarium, and Pythium. In experiment 2, we did see that kind of characteristic fumigation bump where we had numerically higher yields and better plant growth and vigor with our fumigated versus our non-fumigated plots. And we're curious what might be some of the biological factors that are contributing to these differences in growth. So there's a postdoc in Dr. Inga Zazada's lab, Dr. Cedar Hesse. He's looking at soil microbial ecology within these different plots for experiment number 2. So now I want to move on and talk about project number 2. This is alternative fumigation strategies. This is a project that was funded by the Washington State Department of Agriculture Specialty Crop Block Grant Program. So to introduce this project, I want to share that what we found out in the first project, or what we're still learning from other research projects looking at soil-borne diseases, is that standard fumigation with Talone C35 really produces variable suppression of root lesion nematode and other soil-borne diseases. We know from other industries that tarping should improve fumigation efficacy by keeping that fumigant within the soil, allowing it to be effective over a longer window of time, but it's seldom practiced. A lot of times that's being cited to the cost of the tarp. Preliminary trials that have been done with Dominus, soil biofumigant, that is an allele isothiocyanate derived from mustard oil, as well as Talone C35 followed up with an application of Vapam or metam sodium, have been promising. And we've also observed in some previous trials that there are high populations of root lesion nematode in winter cover crop roots. So the question we've had when we see some of these high root lesion nematode populations in winter cover crop roots is, are these winter cover crops that are planted or growing between pre-plant fumigation in fall and then destroyed prior to planting come January, February, so you're ready to plant by March, could these serve as a bridge for root lesion nematode from one planting to the next? So for this particular project, to address some of these observations that I outlined in the introduction there, we have 2 experiments. One was to determine if alternative fumigant products and fumigation practices improve management of root lesion nematode, Pythium, and Fusarium. And in experiment number 2, we're looking at if winter cover cropping practices and modification of those practices could help reduce populations of root lesion nematode from one planting to the next. Due to the interest of time, I'm not going to cover this particular project today, but I just wanted to mention that is something that we're looking at. So for experiment number 1, this is a randomized complete block design. We have 4 treatments, each applied to a single row plot. And so about our total treatment area within a plot is about 700 foot of row. This was replicated 4 times, and the treatments include, the treatments were applied in fall of 2015 and include Talone C35, which we consider our control. Then we have Talone C35 with a high barrier tarp. We have Talone C35 with Vapam, and then we have Dominus biofumigant. What I want to point out is that Vapam and Dominus are both applied with a different rig than what Talone is applied with. So Talone is usually applied deeper in the soil. The shanks are usually about 16 to 12 inches injection depth, and Vapam is about 9 to 9 to 6 inches. So we're getting a shallower layer in the soil when we apply Vapam as well as Dominus. So here are the results from 2016 that I wanted to share. What we're first going to be looking at here is the root lesion nematode populations in the winter cover crop roots. And what we observed is that at the statistical level, we are seeing reduced populations of root lesion nematode in our Talone plus TARP and our Talone plus Vapam treatments. When we look at root lesion nematode populations in June and July, we don't see any differences in their populations at the statistical level. Likewise, when we look at plant growth measurements, primocane number, primocane height, and primocane diameter, we don't see any differences across the treatments. Some of the soil-borne disease data indicate that the root debris were not penetrated by the fumigant, regardless of treatments, and this applies to both large and small roots that Dr. Weiland collected and evaluated. The TARP treatment was initially more effective on Fusarium than it was for Pythium in the bulk soil, but the populations of Fusarium and Pythium were the same by spring of 2016. So we saw a little bit of effect in the bulk soil on Fusarium about 6 weeks after fumigation, but no difference right around the time of planting. And then our fall root lesion nematode populations in the raspberry roots were actually relatively low for this particular site, 12 to 41 root lesion nematodes per grams of root in our September 2016 sampling, which suggests actually across all the treatments that fumigation may have been effective on controlling a root lesion nematode. We'll know more as we transition to next year and we start collecting yield data and continue looking at this particular project. So just to give a summary, I want to mention that these results are still preliminary. We're still in the process of collecting more data on some of these experiments, and we're synthesizing these data. However, what these preliminary results demonstrate the variable nature of fumigation, particularly with Talone C35, and that there are opportunities to improve current soil-borne disease management systems. Right now, Talone plus Vapam and tarping, those look to be the most promising in terms of their ability to control root lesion nematode. But further work still really needs to be done to refine this system. So future work that I want to mention, we're in the process of looking at additional fumigation trials or applying for grants to do additional fumigation trials up here in Northwest Washington. Although it might be a little bit premature, I also would acknowledge that there is a USDA specialty crop block grant pre-proposal that has been submitted by Dr. Nick Grunwald, who is a specialist on Phytophthora at the USDA ARS Center in Corvallis. And some of the goals of this particular research if funded would be characterizing the microbiome and looking a little bit more closely at how soil-borne disease development occurs, as well as looking at more improved strategies or techniques to enhance soil-borne disease suppression. I also wanted to highlight that we are looking at different winter cover crop species or varieties. Up here on Whatcom, there was an experiment initiated this fall with the group where we're trying to identify if there are alternative winter cover crops that could be utilized that are less of a good host or suitable host for root lesion nematode. So remember, root lesion nematode has a very broad host range. It's going to be difficult to find a cover crop that's not a good host for root lesion nematode, but there are some that have the potential to be better than what's currently being used, which might be bobtail or Rosalind winter wheat. So what are being looked at include black oats, tall fescue Jessup Max Q, which has an endophyte in it, so a biological component that should potentially make it less suitable for root lesion nematode. We're looking at Wheeler rye. There's been previous research to suggest that Wheeler rye might be suitable for not being as good of a host for root lesion nematode. And then we have our two controls, bobtail winter wheat provided by CHS and a no cover crop treatment. So I've already at the very beginning acknowledged some of the scientists that are really pivotal in this research. I also want to acknowledge Suzette Galinato. She's an economist at WSU who helps out with some of our economic analyses that we're working on, as well as Mark Mazzola. There's Sean Watkinson, our technician, Chris Benedict, who helps out with extension outreach. Rachel Rudolph is a graduate student in my program that you'll be hearing from next, who has helped out with risk research. Carter DeGraw, Rachel Weber, and Jack Pinkerton have helped out at various stages with data collection. A big thanks to our grower cooperators, especially John Mayberry with Mayberry Packing, Larry Tremaine and Randy Honkoop, as well as Rolf Haugen, the Washington Red Raspberry Commission, Tim Purcell of Trident Agricultural Products, The Whatcom Farmers Co-op, or CH&S now, the Northwest Center for Small Fruits Research, USDA CARE, and WSDA Specialty Crop Block Grant, those last three being our funding agencies. So with that, I want to thank you for your attention, and if anyone has any questions, I'd be happy to